This is a special episode of the Immunology Podcast, IUIS 2025, Day 3. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rao. Welcome back to the Immunology Podcast, where we have conversations with immunologists. We're back with our third episode covering highlights from IUIS 2025 taking place in Vienna. Brenda and I are having a blast attending and getting to meet all of you, but it's not over yet. If you're looking to find out what we're up to at the meeting, follow us on X at @ImmunoPodcast or Blue Sky at immunologypodcast.com. You can also find episodes covering the previous 2 days of the meetings at www.immunologypodcast.com, wherever you find your podcasts. Today we'll be discussing our favorite sessions throughout day 3 of the meeting. So if you were in another session and weren't able to attend the meeting, we've got you covered. We're going to kick off things in just a minute, but before we get to that,
are you looking to improve your T-cell therapy development? With STEMCELL Technologies' new GMP-manufactured Immunological XF, you can achieve robust T-cell expansion with a high yield with viability without the use of serum. To learn more, visit www.stemcell.com/immunocold-XF.
So, day 3.
Day 3. Here
Some
we
day.
are.
Middle of it.
Middle. We're right smack in the middle. Are you having fun so far?
I am.
Is it nice?
Today was a grind, but that's because we did the on the ground episode.
All right. We had a lot of people coming and talking to us.
We had even more than we planned. We had a Jim Allison show up.
We did. That was fun. Those are very nice.
So we had some special guest stars coming in.
You know, by now we're friends with these people. It's just like that. We have Miriam Mirad also, just stopping by to see how we were doing. I like it. I can get used to this.
Yeah.
This VIP treatment. But I think the nicest thing was to get to talk to so many participants and their experiences from all over, people from all over the world. That was pretty nice.
Yeah, I think we got all the continents again, except Australia.
No Australia? Yeah.
No, we got Australia. Sorry, no Antarctica. We got Australia. We got North and South America. We got Europe and Asia, and I think we even got someone from Africa. So Antarctic listeners, we need you here.
I think penguins don't have opposable thumbs. They cannot pipe it, their experiments. So.
There's labs in Antarctica. I'm sure someone's studying the effect of T cells on sub-zero temperatures.
Hey, that's kind of cool. I would do that. Well, or maybe the immune system of like sea lions or something, or the lymphocytes in whales. That would be fun. All right. But let's go back here to Vienna 2025. What, concerns us, which is the talks we went to today and what's been going on. It's been really fun. So many things to do. A really cool talk. So
today was a peak day for me. The beginning and the end were really just strong in the ways of Jason. We started out with mucosal immunology.
Of course. I'm not surprised.
Yep. So one was about the microbiome and gut inflammation from Maria Ressigno in Italy. But then it was about breast milk. So the mother, how the mother, but also gut inflammation. So mother IgA protects the baby's epithelium while your mucus comes online, right? Like when you're born, you don't have a lot of mucus, so you need this IgA. And then sometimes everything goes to hell, and then you get necrotizing enterocolitis. That's real bad. So they know that antibiotics are related to this somehow. So they're showing that antibiotics in dames and the mommies deplete IgA in both the moms and the pups.
What?
Yeah. Well, so if you give mom antibiotics.
Oh, antibiotics. I understood antibodies. It's like no. Antibiotics. Yeah. Okay.
You can do that too. And then they really showed that it's all maternally derived. Every IgA in itty bitty baby mouse is from mommy mouse.
How generous of her.
Mm-hmm. And there are, but more wacky than this is that when you do this, and then so you're now giving them an IgA depleted and the mom has less microbiota, which she also gives to her baby, you affect the plasma cell function of the babies permanently.
Permanently? That's scary.
Mm-hmm. And then that leads to more susceptibility to intestinal inflammation like NEC.
Wow. I mean, I don't want to spoil it because we've heard a lot about, antibiotics and detrimental effects on our immune capacity. So this is just adding more to the fire.
Yeah, that'll be for later. I told you it was a good day for Jason. Yeah. But then, that was pretty neat. Then we had someone who was a tour de force in IBD, host microbiome interaction for the maintenance of intestinal homeostasis from Kiyoshi Takeda. So they did a lipidomic analysis in IBD patients and found patterns of disruption in Crohn's patients, but not colitis patients, identified a specific lipid protein that is converted by PLA from phosphoserine to lysophosphorserine. It's a specific one. And there's more E. coli with this enzyme in Crohn's disease patients. And then they were able to demonstrate in mice that exacerbated T-cell colitis, and that if they got rid of the receptor for it, the problem went away. And then they had a separate talk that I was thinking they were going to relate to the first one, but I couldn't follow if it ever actually related back to the first part of what they were doing, looking at deep profiling of human T cells in a tour de force, and really identifying resident memory T cells that have a lot of overlap with NK cells and function through interferon gamma and a lot of RUNX2 and BHLHE40, and these are little bad naughty T cells that help drive disease. But it's like a T cell that's being like an NK cell, which, as we were discussing with our friend Dr. Allison today, all immune cells are just a smear, or as I like to put it in the microbiome world, a skid mark.
The skid mark of immunology. Yeah. Yeah, in the end, these neat categories are man-made for our very our man-mind understanding, our human mind understanding, but they're not real. The human borders are not real. They're constructions made by us to make sense. Okay. Anything else from your morning?
That was it, because then we were in interviews.
Indeed, that was intense. Well, I did have time also to go to a couple of talks before our marathonic sessions. I did have a little accident on my way to the conference. I slipped on a glossy brochure on the floor. Can you believe how silly that sounds? And I made a hole in my pant and in the knee. It was very sad.
And in your knee.
And in my knee, and I also made a hole in my knee. So not very fortunate of me. But well, doesn't matter. Went back, changed pants, but that means I missed a bit of the first talks. But I did get to kind of get the end of, or like most of the talk from Lara Cook from, I think, Melbourne University. And she had a lot of studies looking into patients that were, severely ill after, for example, COVID or after trauma, and studying why is it that these patients, after they kind of recover, they're still to some extent immunosuppressed, and what is the effect that lingers after the kind of the apparent recovery of the patients that is actually negatively affecting their health. And so she's talking. So some of the things that they looked in, I think most of their interest was around phagocytes and dendritic cells. So phagocytes like neutrophils and monocytes, and basically they show that there's differences in the phenotypes, and there's this particular impaired dendritic cell type that it's CD11B negative, SIRP1 alpha negative, that seems to be an early marker of this immune paralysis that they describe. So that's really nice, and then she was also showing some other smaller things, and she had this cool human lung organoids, and she showed this really nice pictures of ciliated cells, and they're super funny because they have like this little kind of, oval shaped with a cilia on one of the sides, and it looks like pineapples. So I thought it was really fun. So after her talk, was also Matteo Iannacone. And we have covered his recent paper recently in the podcast. He is a PI in Ospedale San Raffaele in Milano, in Italy. And he basically shared with this story that we already covered, but I thought it was so good, and it was a really nice presentation about the importance of CD4 kind of help, but in the priming. So in the case of the liver, CD8, so it is in the context of HBV infection, chronic infections, and this exhaustion, or this kind of, this dysfunction that you see on CD8 cells. And one of the things he starts with is showing that, or kind of reminding us that in the liver, because the epithelium of the liver is so thin, T cells can directly interact with the parenchyma and can be CD8 cells can already kind of be primed by the parenchymal cells that are infected with this HBV directly, but this is actually not a good thing because that usually results in kind of this dysfunction and of this CD8 HBV specific CD8 cells. And this can be reverted with the help of CD4 cells. And so this dysfunction can be prevented. And this is through the interaction and this kind of triad of CD4 cells, CD8 cells that are put together by resident macrophages from the liver, which are known as Kupffer cells, and that this really the presence of these cells, and not dendritic cells, so like CD11 or CD11C dendritic expressing dendritic cells, but rather CLEC4F, I think, expressing Kupffer cells, are this key cell type that is in between the CD4s and the CD8s. And that to some extent, this is mediated by production of IL-27 by these Kupffer cells. And that is what is mediating this help. And it's very important for the establishment of robust antiviral immunity in HPV infection. And so then we had the whole we have our guest here in the studio, and maybe I'm just gonna talk about two talks that I attended afterwards in the afternoon before the plenaries. So I was a super nice session about kind of visualization. I forgot the name of the session, but it's like visualizing the immune system. So it was like really cool microscopy and using fluorescent. So the first talk was from Gabriel Victoria from Rockefeller University. So he had this model that he studied germinal center, kind of how germinal center B cells, they kind of start diverging from each other, and he used this mouse model. I missed the really first part. I think it's a mouse model that's called brainbow mice. I think they're usually originally used for neurobiology studies, so you can differentiate different neurons, because then you have like stochastic expression of different colors, different fluorochromes and fluorescent proteins. And so that allowed him to study individual clonal expansion of germinal centers. So if a cell if you see a cluster of cells that are of B cells that are all the same color, then you can kind of infer that they all came from the same original cell that expanded in response to antigen and it became a kind of a germinal center. And so he used this to really study, to isolate germinal center cells and really analyze them in single cell sequencing and looking at their B cell receptors and the antibodies that they're making, and understand in a way the phylogeny of all the different relationships between the B cells and how germinal centers work. He compared affinities and the sequences of the CDR3 region, and basically he concluded a couple of things I thought was really good at the end of his talk and showing what he learned. And what he learned is that, so when he looked at the kind of mutations that that happen during germinal center reactions is a normal thing. The B cell receptors get mutated in the hopes that you'll find the one that has higher affinity, that will make better antibodies. And so what it shows is that this process is actually pretty inefficient, and germinal centers miss most of the affinity enhancing mutations. They're actually pretty rare. And the focus and the most common mutation that happen, and this is not surprising when you think about it, but it's kind of are the ones that are more easily generated, if that makes sense. So there are some types of mutations, that some locations and some particular changes that are easier and more frequent, and that's basically the ones that you are going to see in this population. There's not a lot of the kind of selection pressure is actually not as much as you would it's less than you would expect. And although germinal centers are intolerant of loss of affinity, so cells that really lose affinity as a consequence of mutation will become less and less prevalent. But sometimes it happens that you will have affinities that are stuck in a local max. So you might be able, if you had a less affinity, you could maybe be halfway through a mutation actually gives you a better affinity in the long term, but that that transition is really hard and it's really hard to get. And so you don't see that very much. And also that clonal bursts are not the primary drivers of affinity maturation. And this tells that the germinal center selection, it's relatively inaccurate. It's not such a driving force. So it is biased in general in the right direction, but it's not like you're going to have this massive, one clone that is slightly better than the rest is going to completely obliterate the population. But we do have kind of a general movement towards better affinity and better receptors. So this is really cool because all these things are really hard to kind of study. So I thought his system and all, he was very careful. And so he had this really, really nice approach to studying this really complex process. And they shared here their bioarchive. They published this on bioarchive. So really, really cool. I think I'm going to try to take a look at it online because I thought the system was really nice. And then we had also a really cool talk from Ashley Mosman from Duke University. And it was a bit of a... at the end, I was a bit worried because it was about this brain-eating amoeba, this Naegleria fowleri, that is this amoeba that has the ability of infecting the brain through the nasal epithelium, and so the nerves that innervate the nose, and then they go directly to the brain. And so the question is, how is this amoeba allowed to because once it establishes itself, it just starts eating from the olfactory bulb all the way into the brain. And the question is, why are we allowing this, quote unquote, to happen? And so basically what it shows, they did a lot of studies, really cool microscopy, a lot of this with really cool microscopy. And they show that although, for example, you will see detection by neutrophils, but it looks like one of the main limitations to identify and kill this amoeba is that it requires serum-derived complement deposition. And that's really hard to get when you start getting deep into the brain and into the olfactory region. So you don't have access to this complement. And that allows the amoeba to just kind of come in unchallenged. And once it's in the brain as well, they don't do damage fast enough to allow serum complements from the serum to get into there. So that's how they kind of are always a step ahead of neutrophils, who would be the best cell to find this amoeba and kill it with all those nasty things that they can produce. So it was pretty interesting. It reminded me that this amoeba exists that can, infect your brain from your nose, and that make me very scared. And that there's nothing we can really do about it. But at least we understand why it makes it all the way there.
I mean, it's a straight track. There's nothing in between. Your nose is your brain.
Kind of, yeah. So I thought that was very nice and very scary at the same time. And I think I'm just gonna say. Talk briefly about the keynote from Ruslan Metitov, and then you can finish our conversation very quickly. So I just want to say also a very cool story from Ruslan Metitov, and this was talking about the differences between having, specific germ-free pathogenic, so SPF mice versus pet shop mice with a kind of very diverse wild type kind of microbiome, and the effects on allergic sensitization that it's different between SFP mice and pet shop mice. And basically what he shows is that one of the mechanisms by which, so if you can make a black 6 mouse allergic through, for example, against OVA, and you can generate a strong population of Th2 responses, and the mice can die of anaphylactic. And this is not something that you will see if you have like adult pet shop mice. And one of the mechanisms of this protection that he suggests is that in mice that have, wide exposure to many antigens, that have many, so he shows some pictures of the lymph nodes, like already in the steady state, they are like twice the size of like mice, like lab mouse. So they already these mice clearly have seen many antigens. And some of those, what he calls the antigenic space has already been taken by responses that have, of different types, Th1, Th17, some Tregs, and they are already taking up, these responses are already kind of taking up some of this antigenic space. So then if you try to sensitize the mice against an antigen, then there's less space for this to become a full-blown Th2 response. Whereas clean mice, so quote unquote, they have all that space. It's all free because they have not encountered any similar antigens or any, potential cross-presenting or cross-reacting antigens. And so therefore, if you induce a Th2 response, you have all these TCRs, all these B cells, they're all ready to be driven into a Th2 response and make the mouse allergic. So I thought that was like a really cool brain kind of, very tickling idea. It's really, really nice.
Too clean is too clean.
Yeah, too clean is too clean.
That. All right. So last but very much not least, Lawrence Vogel gave a tour de force on the microbiome. And so for the next 3 hours now,
you got 5 minutes.
So she saw some cool stuff. She showed that dysbiosis is related to poor outcomes in cancer. Yeah. And that antibiotic use, which can drive dysbiosis, leads to poor outcomes in cancer, especially with immunotherapy or CAR T therapy. They were able to validate MAdCAM1, serum soluble MAdCAM1, as a measure of dysbiosis, which was validated in the Javelin Renal 1 trial, which is in revision now. And she showed some of that data, which is really cool. So they were able to come up with a soluble marker of dysbiosis instead of a, sequencing. That's really awesome. She also noted that vaccines fail in 25% of people, they work, but they don't have the T cell immune response in 25% of people. And this was true in healthcare workers with COVID and in experimental vaccines for melanoma and lung cancer. HLA genotyping in the COVID cohorts related no relevance to that, but stool metagenomics did. So your microbiome also affects your vaccine ability. And then they showed specifically the loss of Akkermansia, which is a kind of good bacteria, leads to loss of CAR T cell maintenance in a mouse system. So she talked about some of that data with human data. And they also showed that low-dose ileoradiation reduces resistances to CAR T therapy, and they think that's microbiome related, but not sure why. Showed that FMT potentiates immunotherapy and CAR T therapy. And then she has a company that had developed a specific Akkermansia strain that's now looking at trials and show that they could, do it and colonize, which is the first step to any microbiome. Like if I give you the drug in your mouth, does it stick? And they showed that, but that they can use it like FMT to potentiate immunotherapy. And, just the whole point being that there's a whole microbiome immunotherapy / CAR T cell therapy story that's evolving. It's still early days, but could be very cool.
Yeah, that's super impressive. And it's really exciting. If we could really improve response to therapies, the number or the results that she showed of all these clinical trials were very impressive.
Dr. Allison was saying how it only works on 30% of people and he wants to see it 50 or 60. Maybe you just need an FMT ahead of time.
Yeah. That's really, really cool. And so much potential, because that's something that you can really, if we know something that you can not easily, but you can manipulate. So it's if only people were working on, microbiome products to restore.
If only there were companies that worked on that.
Yeah, if only.
All right.
This was fun.
Well, that brings us to the end of our latest IUIS 2025 episode. We're about to go replete our microbiomes with some dinner. But while we're doing that, don't forget to follow us on X at @ImmunoPodcast or Blue Sky at immunologypodcast.com to find out what we're up to at the meeting. Check back here tomorrow for another episode recapping day 4 of the meeting. See you then.